Yeah, good afternoon, Dhruvi Patel. Good afternoon. Okay. So, today's our topic of discussion is NEET last year's question, and the topic is the prokaryotic as well as the eukaryotic transcription. All right. So, first of all, we discuss prokaryotic transcription, and then we discuss eukaryotic system. Just a minute. Okay. So, the first question is, yeah. So first question is, in bacteria, heat shock response is primarily controlled by. This is the question asked in 2015, June 2015. So the choices are the sigma. Just a minute. Okay. The choices are the sigma S, sigma 32, sigma E, and sigma 70. See, we know very well that the holoenzyme, I mean RNA polymerase, holoenzyme, it consists of sigma factor plus core enzyme, right? Along with question, we discuss a bit more things so that we can be able to solve the many more questions. So this is the holoenzyme. Holoenzyme, it consists of sigma factor plus the core enzyme, right? See, the most commonly used holoenzyme, that means this is nothing but we are talking about RNA polymerase, isn't it? Okay. So this RNA polymerase, it mostly the RNA polymerase used, or the holoenzyme used in the transcription, they are having sigma 70. So sigma 70 is the most common kind of sigma factor which is used by the holoenzyme, right? But here they are talking about the heat shock response, right? During the heat shock response, we heard about the heat shock response, HSP proteins, right? HSP proteins are there, heat shock proteins are there. During the, any adverse condition, heat shock proteins are expressed. And for heat shock proteins, the sigma 32 actually, the sigma 32 is the sigma factor, right? Sigma 70 is the most commonly found sigma factor. This is most commonly found, right? This is most commonly found sigma factor. And in the heat shock responses, so sigma 32 it expresses. Here in this figure you can see. So this is the sigma 32 protein, right? And this sigma 32 along with the the core enzyme, then they form the holoenzyme. Okay, so the correct answer here is the sigma 32. All right. Yeah, Dhruvi, you are correct. Yeah, good. Next question, which of the following statement is not correct? So be careful during these questions. Not correct. So together with the protein, RNA provides a site for the polypeptide synthesis. See what they are asking. which of the following is not correct, right? So together with protein, RNA provides a site for polypeptide synthesis. Okay, they are talking about not correct. Okay, so together with protein, RNA provides a site for the polypeptide synthesis. Okay, so all DNA molecules are unbranched polymer of the nucleotides. Yeah, this is unbranched. DNA is unbranched polymer, so this is correct. Okay, DNA is synthesized in 5' to 3' direction, while the RNA synthesis takes place in 3' to 5' direction. No, not at all. See what happens during the polymerization. We know very well during the polymerization, whether it is DNA synthesis or RNA synthesis, this 3' OH, okay my dear friends, this is 3' OH, and it makes a nucleophilic attack on the alpha phosphate. This is alpha phosphate, right? So it makes a nucleophilic attack on the alpha phosphate, and finally bond will be formed here. The ester bond, phosphodiester bond will be formed, right? So this statement is actually wrong, but last statement we have to read. A tRNA anticodons may pair with the more than one codon. This is also correct. Okay, so here the wrong answer is 3. Okay, all right. First, together with the protein synthesis, RNA provides site for the peptide. Together with proteins, rRNA, it provides the site for the polypeptide synthesis. This is correct. Second number is correct. So third number is the wrong answer. So this is the correct answer here. All right. 2C. Yeah, 2C is correct. Yeah, very good. Rupesh sir, good afternoon. I'm sorry, actually I was late due to some technical issues. Okay. Now, third number question. Sigma unit in E. coli RNA polymerase does not, again the twist is does not, right? So initiate transcription and fall off during elongation. Yeah, they sigma factors are responsible for the recognition, right? Recognition, they scan for the Tata sequence as well as the other sequences of the promoter, and they join over there, right? So they initiate transcription and fall off during elongation. Yeah, this is correct. Once the transcription initiation enters into elongation, then there is no use of sigma factor, isn't it? So suppose this is I'm making, that RNA polymerase, and this is suppose sigma factor. So sigma factor, they scan for the Tata box and other sequences here in the promoter, right? And once they recognize, they start the transcription. During the elongation, they fall off. So this is correct answer. Second, increase affinity of the core enzyme to the promoter. Yeah, of course, increase affinity. They are having increased affinity of the core enzyme, right? Sigma factor. Then only they can form the holoenzyme. All right, to the promoter. So this is correct. So it binds to DNA independent of the core enzyme. No, this is not correct. So sigma factor always binds to the DNA along with the core enzyme. All right, otherwise it won't be able to bind over there. So this seems wrong here. Fourth number, ensures specificity of the transcription by interacting with the core enzyme. So this is correct. So second number is the wrong answer, right? Yeah, Anjali, you are saying 3. Yeah, you are correct. Anjali is correct. Komal Patel 3. Yeah, good. So this is correct. Okay, third number is the correct answer. So all of you are correct. Okay, very nice. Now 4 number question. Which one of the mechanisms used by the bacteria for adaptation to changed environment is altering transcription of their genes? Okay. So in this regard, which one of the following responses not found in the bacteria? Again, they are asking not found in the bacteria, right? Okay. Due to the change in the environment, right? Change in the environment. Normally they, change the transcription pattern or transcription of the, different genes, right? So in that regard, they are asking which of the following is not correct. So first is a gene with two different promoters for expression in different conditions. Yeah, this is correct. If two different promoters and two different genes are there, then what is the problem? So they certainly express, right? This is correct. Use of the different sigma factor for the transcription of gene. Yeah, of course. In the first question we talked about that sigma 70 is the general, very common sigma factor, right? Very common sigma factor. And like sigma 32, they normally express in the heat shock condition, right? So they are expressed during the heat shock condition. So many more kind of sigma factors are there. So this is also correct. So 2 number is also correct. 3 number, expression of alternate alpha, beta, beta dash subunit. Yeah, this is also possible. During the changing environmental condition, this core enzyme, what we talked about in the first question, that they are made up of alpha, beta, beta dash, gamma and omega, right? Alpha, beta, beta dash, omega, omega dash, right? And then sigma. This is sigma, suppose, right? Yes, sigma. So of course this may also be possible. So they expresses different kind of beta, beta dash. Yeah, 4D, Dhruvi. Yeah, 4D. Yeah, you may be correct. And then 4th number is expression of anti-sigma factor. No, anti-sigma factor, this is nothing like that actually. And they want produce anti-sigma factor. When sigma factor is not there, then how they can bind to the promoter, right? So this may be the wrong statement. That's what they are asking. Which one of the following responses are not found in the bacteria? So here the correct answer is D. Alright, you are correct, Dhruvi. Okay, good. Okay, next question. I think you are some portion you are not able to see here, so we discuss in the next slide, right? So the question is the row fact. This is very long question actually. This is a very long question, but believe me, this is really easy question. Whatever things I have written here, this is just for the information. I am telling you, right? So don't get panic by looking at the long question. This is a really simple question. Whatever I have written, this is just for the information, right? So the question we should read first. So rho factor is involved in the termination of the transcription in prokaryote. This is just information, right? We know very well that rho factor is responsible for the termination, okay? Then genetic manipulation indicate that, don't worry if you are not able to see the back written things, right? I'm explaining. So genetic manipulation indicate that rho dependent, see, two different kind of terminations are there. One is called rho dependent and one is called rho independent, right? Rho is a protein, right? Rho is a protein which normally, works during the, or is responsible for the termination of the transcription. Yeah, I think it is now visible. I'm getting a problem in the internet connection actually. Very weak internet connection today is there. It disconnects in between actually. Okay. So this is the question then. Then that sequence is, that synthetic sequence is poor in the C and rich in C and poor in G, right? So that sequence, that synthetic sequence they are talking about is rich in C and poor in G, right? So first of all, we try to understand what is actually rho independent and rho dependent termination. So here the rho dependent termination. So rho dependent termination means rho protein is there, right? So rho is the protein which is actually less characterized protein. This is the first point. And rho is nothing but a ring shaped hexameric protein. Ring shaped, they are hexameric protein. and it normally binds to the single stranded RNA as they are saying in the question also, right? Okay, and then another important thing about the row dependent termination is they are having ATPase activity, right? And what they do actually, with the help of this ATPase activity, they pulls the RNA from the template, right? Or from the template or from the RNA polymerase, right? And two different ways are there for the row dependent termination. One is they bind to the root site, something which is called RUT site, row utilization for termination. This means RUT, row utilization for the termination site. And there, this is 40 nucleotide long site on the RNA actually, right? And they don't form the secondary structure, hairpin loop, as we have seen in the question also, they are saying, okay? And they are rich in the C terminus. Actually, they are talking about the row dependent termination. And another important thing about the, another important way about the row independent or row dependent termination is that row fails to bind at any transcript that is being translated, right? If it is bound to the ribosome, I mean that 40 nucleotide site, root site is bound to the ribosome, then they are not able to bind over there. So in bacteria, where we know the transcription and translation is coupled, right? So there it is very difficult for the row dependent proteins to terminate, right? So now we come back onto the question, right? So the choices are the row factor attaches to the nascent RNA at the recognition sequence and then migrates along the RNA in 5' to 3' direction until it encounters the RNA until it encounters the RNA polymerase paused at the termination site and disrupts the transcriptional complex by acting as the. So actually what we learned here in the row independent termination, this is the correct statement about what they are saying in the description of the 5th number question, right? So this seems correct answer, but second we have to also read. So row unwinds the RNA DNA duplex formation transcription bubble, thereby releasing the RNA transcript. No, not at all. They won't form the RNA DNA duplex forming. Transcription bubble they are not forming. Then third choice, row factor attaches to the RNA at its recognition sequence, while the RNA in the RNA, no, that is not the case also, we have seen, right? And fourth number, there may be the other factors and hence row factor does not need to unwind the RNA DNA hybrid to release the no, this is also not the case, as we have seen in this row dependent termination, right? So here this first number is the correct answer. Whatever things related to the row dependent termination they said in the description of the question 5 actually, right? That is written here. I have written some more information about the row dependent termination. But apart from that, we should also have the knowledge about the row independent termination. So this is the row independent termination. You know, as opposed to the it is opposite to the row dependent termination. So in this row independent termination, so here the hairpin loop formation is there, right? So RNA is synthesized in a way so that they are complementary to each other. The portions are complementary to each other. That's why they're forming the hairpin loop, right? And due to the formation of hairpin loop, the RNA polymerase, they falls off from the RNA DNA complex, right? So this is about the row independent termination. So question was about the row dependent termination that we have seen, okay? So now the third question. So actinomycin D, it inhibits the process of transcription. So actinomycin D is the antibiotic. It inhibits the process of transcription in both prokaryotes as well as the eukaryotes, right? Okay, so both prokaryotic and eukaryotic transcription are affected, okay? following statements are made about the actinomycin D mediated inhibition of transcription. So this is important actually. So 4 statements are there, and we should read the statements. Actinomycin D inhibits the transcription from a double-stranded DNA template by either E. coli or by yeast DNA polymerase. Of course, actinomycin D act on both prokaryotic as well as the eukaryotic transcription. Okay, so this is correct. Yeast is eukaryote and E. coli is prokaryote. Okay, so this is the correct statement. Okay, so now from here, okay, yeah, of course. Then second choice, actinomycin D, it inhibits, so first number is correct, it seems correct. So actinomycin D inhibits transcription from a single stranded RNA template by eukaryotic viral RNA polymerase. No, not at all, not viral RNA polymerase. So this is wrong. Actinomycin D inhibits transcription from a single stranded 5x174 DNA template by E. coli RNA polymerase. No, not at all. They inhibits prokaryotic as well as the eukaryotic transcription, right? How they inhibit, we'll discuss. So fourth statement is actinomycin D inhibits transcription from double-stranded RNA template by eukaryotic RNA polymerase. Not only the eukaryotic RNA polymerase 2. So this is also not correct. So A seems correct. A is all right here. A is all right. But before selecting the answer, we should look at this table so that other questions can also be addressed. So these are the different inhibitors. Some of them are antibiotics, some of them are not antibiotic, other compounds, right? And these are the affected organism. So this is, so first of all we talk about the actinomycin D. So this is the actinomycin D, right? This is actinomycin D. So they work on both prokaryotes and eukaryotes, and they inhibit RNA polymerization. Okay, so that the question was, right? But apart from that, here other important informations are there. So another compound which is alpha amanitin. This is the product from fungus Amanita phyloides. Amanita phyloides. This compound is isolated, and this works on the eukaryotes only, and they inhibit RNA polymerase 2, right? Mostly, in most of the cases, RNA polymerase 2, then RNA polymerase 3, and very less amount of RNA polymerase 1 has also been affected. Here no effect it is written, but very less amount of RNA polymerase. This is least effective, we can say, right? And RNA polymerase 4. So Amanita, they asked 2-3 times in the NEET examination. We'll discuss in the next question. Then you can also keep in the mind heparin. So heparin, it acts as prokaryotic transcription inhibitor in the prokaryotes, right? And inhibition of the transcription in vitro. Apart from that, rifampicin, they are prokaryotes only, act on the prokaryotes only, and inhibits the chain initiation. At this stage they initiate, they inhibit. Then okay, so then last one, this is the anthracycline. So both prokaryotes and eukaryotes, and this is again the intercalating and stops the RNA polymerization. So this table is very important in short, right? So here again we come to the question. This is the structure of actinomycin D. This is actinomycin D, right? This is actinomycin D, and here you can see they are having alpha ring and beta ring, right? One ring is called alpha ring and another ring is called beta ring, and these rings are, suppose this is the alpha ring, so this will become beta ring. So they intercalate in between the bases. See, this is the alpha ring, this is the beta ring, right? And that's how they inhibit the transcription. So here the correct answer is 1. 1 is the correct answer, right? Only A. So actinomycin, it inhibits the transcription from the double-stranded RNA template by either E. coli or the RNA polymerase of the yeast, eukaryote. All right. Now certain questions from the eukaryotic transcription. So again, the thing we discussed just now, which is alpha amanitin. So alpha amanitin is a fungal toxin which inhibits eukaryotic RNA polymerase. This is correct. 3 eukaryotic RNA polymerases showed differential sensitivity to the toxins, right? So which one of the following order is correct? High to low they are asking, okay? So what we discussed, RNA polymerase 2 is affected mostly, right? Sensitivity of the RNA polymerase 2 towards the alpha amanitin is much more, okay? So RNA polymerase 2 is highest, then RNA polymerase 3. So the choice may be this one, RNA polymerase 2, then RNA polymerase 3, and RNA polymerase 1 is least sensitive, okay? So this would be the correct answer. This is important question. And information wise question, this is correct. This is very important, okay? Again, in 2011 they asked, alpha amanitin inhibits. If only one choice is there, then we have to write this one, right? So only RNA polymerase 2. Actually, this is not the case. The case is RNA polymerase 2 is much more sensitive than RNA pol 3 and then RNA polymerase 1. Okay, this is RNA polymerase. Okay, so this is the order in which it acts actually. So here the second answer is correct. Okay, not all RNA polymerases, right? Because this is almost not sensitive. Almost this is not affected. RNA polymerase 1 very less sensitive. So if we have to here, all of the above answers are also there, but RNA polymerase 2 is highly affected, very sensitive to alpha amanitin. All right. Okay. Then next question, in eukaryotes, precursor of microRNA. See, 3-4 questions they asked, and we have one table, and by looking or keeping that table in the mind, we can solve any questions of the different kind of RNA question, right? So in eukaryotes, microRNA, that is called miRNA, and small interfering RNA, siRNA, are usually synthesized by. See, before getting the answer or before discussing the answer, we should have this table in our mind or this flow chart in our mind so that it can be answered very easily. See, I am writing something here. This is RNA. So two different kind of RNA has been categorized, coding and non-coding, right? And this mRNA is actually, this is actual coding RNA, and this can be kept into the non-coding also, right? In some classification they can be kept into the non-coding also, right? So here they are talking about the non-coding RNA. So this is snRNA, right? Then SNO RNA, microRNA and siRNA, right? Almost all of them are synthesized by RNA polymerase 2, right? RNA pol 2, right? They are synthesized by pol 2, RNA polymerase 2, right? And here the function is written. This is very important. Really, this flow chart is very important. So snRNA, they act in the processing of the pre-mRNA. Just keep in the mind. You will see all the questions they asking in the NEET examination can be solved by this table, this flow chart. SNO RNA, SNO RNA, they are responsible for the, they are responsible for the processing of rRNA, right? Here it is mRNA, pre-mRNA, and this is for the pre-rRNA. Okay. Then micro or miRNA, this is regulation of the gene expression. They are involved in the regulation of the gene expression, keep in mind. And siRNA, we know the antisense RNA, they silence or gene silencing, right? This is the function. Okay. And almost all of them are synthesized by the RNA polymerase 2. And if you talk about the coding RNA, then mRNA, rRNA and tRNA, right? So mRNA concentration is approximately 2 to 3% of the total RNA, right? Not much, okay? But the rRNA is 95% or more than that actually, okay? So now you come back to the question. So question is, in eukaryotes, precursor of microRNA, miRNA and small interfering RNA are usually synthesized by. So what we discussed, they are usually synthesized by RNA polymerase 2. Okay, here almost all of them are synthesized by RNA polymerase 2. Okay, all right. So you keep in mind first thing. Okay, RNA polymerase 2. Here the answer is RNA polymerase 2. All the different kind of non-coding RNA are almost synthesized by RNA polymerase 2. Now, keep that table in mind. Which one of the following statement is generally true about the RNA polymerase 2? So which one is the correct? Right? Generally about the RNA polymerase 2. So here it is dedicated to transcribing RNA from the single transcription unit, generally a large transcript, which is then processed to yield the 3 type of ribosomal RNA. Not correct always, right? This is not correct. More than 3 different kind of RNA they can synthesize, right? And if we talk about the eukaryotic system, I mean regarding the RMT, rRNA, mRNA and tRNA, so only the mRNA is synthesized by the RNA polymerase 2. I mean if we are talking about the coding RNA, okay? Then second choice, it transcribes varieties of small non-coding RNAs which can express in all cell types. Okay, see till here it is correct. Almost in it transcribes varieties of the small non-coding RNAs. Here till here it is correct, which are expressed in all type of cell. This is not wrong. This is not correct. This is wrong actually. In prokaryotes they are not able to synthesize. Okay, all right. Third number, it generally synthesizes various type of mRNA and small non-coding RNA. So this is correct. I mean this seems correct, right? Fourth number, it is exclusively involved in the synthesis of rRNA, and no, not at all. Okay, RNA polymerase 2, they synthesize mRNA. So here the correct answer is third. Third number is the correct answer, right? So this is the third. I hope you are getting. So all the different kind of non-coding mRNA, non-coding RNA are synthesized by RNA polymerase 2, but not in all type of cells, right? They are, majority of them are in the eukaryotic cells. Okay, so again you keep this table in the mind, so you can solve any kind of question. I mean variety of questions they are asking in the NEET examination, right? Okay. So now we move on to the fifth question. Again same table you can utilize to answer this question. Which of the following are not transcribed by RNA polymerase 2? See, miRNA and some snRNA can be transcribed by the RNA polymerase 2, we have discussed, right? Then mi and SNO-RNA, this is also transcribed by the RNA polymerase 2, alright, we have seen. Then mRNA and SNO-RNA, this can also be synthesized. I mean mRNA to hai hi, apart from that SNO-RNA can also be synthesized. tRNA cannot be synthesized by the RNA polymerase 2. So the correct answer is 4. Again I am showing you this table. This table is very, very important, and you can make a snapshot of this, and certainly you will get answer of many more questions. Okay, all right. Now we move on to the other question. Again see, 2014 they are asking. They asked small nucleolar RNA, right? SNO-RNA, right? They are talking about SNO-RNA used to process and chemically modify rRNA are called, right? Small nucleolar RNA used to process and chemically modify rRNA, right? Okay, see, again you come back to the table. So what they are asking, the small, not SNO here, you just cross it. So which of the following RNA is responsible for the modification of the rRNA? They are talking, right? Which of the following are responsible for the modification of the rRNA? So you come back to the table and see here. See, so which of the following? So we have see SNO-RNA here. See, SNO-RNA, processing of the rRNA. SNO-RNA is responsible for the processing of the pre-rRNA into the rRNA, whereas the snRNA, they are responsible for the processing of the pre-mRNA, right? So you can make different kind of mnemonics by using this table, because many questions they are asking. Okay, so here the answer is SNORNA, snow RNA, right? Here the answer is third number. Okay, so the snow RNA is responsible for the modification of the pre-rRNA, right? That's what they are asking. Okay, again, see, again many times they ask this question. So certainly they can ask maybe in the B section or in the C section. It depends upon the complexity of the question. Again, see, in list below are 5 different type of RNAs produced in the cell column A, and the function is there in the column B, but not same order. Okay, not same order, so we have to match here, right? Okay, so we can match here. See, just we have had discussion about the snow RNA, right? SNORNA. So snow RNA they used to process and modify the pre-rRNA. So anywhere pre-rRNA is there, so here rRNA is there. So this may be snow, right? Okay, miRNA again. You just refer to the table. I am showing you table again so that the newcomers can see this table. This is very important. I mean flow chart or table, what you say. So here we have seen the snow RNA is responsible for the processing of the pre-RNA. snRNA for pre-mRNA, I mean processing of the pre-mRNA. So that is also there in this matching. So see, this is the snRNA. So snRNA is responsible for the pre-mRNA. So here the third number is there, this one, isn't it? Then siRNA, the siRNA is there. Now you can write the answer in the description, in the chat box. So here the B number, siRNA, they are responsible for directing the degradation of the selective mRNA, right? You can again refer to the table. And miRNA, so they are responsible for the regulation of the gene expression by blocking the selective mRNA. So this is second number. Okay, everything is written in that table. If you look at this table, everything is here. This is very simple table, very simple flow chart, but everything is written here. Okay, so the correct answer would be 1 3, right? 1 3, then B1, so B1, then C2, yeah, C2, and D4, yeah, D4. So this is the correct answer. All right, yeah. All right, okay. So now we move on to the next question. Yeah, this one. This is very interesting question. You can see here. Yeah. So genetic studies demonstrated that TBP mutant cell extracts are deficient in the transcription of gene from all 3 promoters, like class 1, class 2, and class 3. If TBP mutants are there, if some mutations has been made in a cell in the TBP gene, then it has been seen that the transcription of all the 3 genes has been affected, right? Following statements describe the characteristic feature of the TBP, right? So first, TBP is considered as an universal basal transcriptional factor. This is correct actually. TBP is the universal transcription factor. They are present in the all eukaryotes, all prokaryotes. So TBP is considered as universal basal transcription factor, right? This is correct. Okay. TBP is not required for the transcription of archaeas. No, they are required, right? They are required. Okay, so this is not correct. So TBP is involved in the recognition of Tata box. This is correct. TBP is involved in the recognition of Tata box because this is Tata binding protein. Okay, TBP. Then fourth number, TBP operates at all promoters regardless of their Tata content. This is also correct. This is also correct. Okay. TBP operates all promoters. Okay, I'm sorry, this is wrong actually. TBP operates to all promoters regardless of that. No, Tata should be same. This is a conserved sequence. So Tata should be same. So this is also wrong actually. Okay. So now the question is, which of the following combination is not correct? Right. So A and D is wrong, but A is correct. Right. So this is not the answer. C and D. Okay, D is wrong, but C is correct. So again, this is not the correct combination. Here in the third number, B and D, B is wrong and D is also wrong. So this is not correct combination. And fourth is A and C. So A is, here the A is TBP. Okay, A is correct and C is also correct. Alright, so this is correct. So correct answer here is third number. So third number is the correct answer. Alright. Yeah, Anjali, you are correct. Anjali V, you are correct. Third number. Yeah, I'm sorry, actually today the session was interrupted 2-3 times by poor internet connectivity. I'm sorry for that, but please bear with me. Now the 9 number question. Highly specific inhibitor that targets the photophosphorylation activity of TF2H is added to an in vitro transcription reaction. Which one of the following step is most likely to be affected? See, again, this is good question. This can be considered as good question. Yeah. So can you answer? So highly specific inhibitor that targets TF2H. See, this is the transcription factor, right? And this transcription factor comes at the end of the transcription initiation, if you remember. So this comes at the end, and this is the largest transcription factor, TF2H. It comes after E, and then H comes, right? Okay, we have some figures to explain this, right? But here we can search out the answer, right? So what is the problem? So which one of the following step is most likely to be affected, right? So see, okay, getting answer is not a big deal, but we have to just see this one, okay? Then we can get good concepts how to solve this question and related question. Again, other questions are also there with this figure and table. So see, this is the Tata box and this is the TBP, right? And TF2, this is TF2D, right? So after D, then A will come, right? Then TF2B will come. So you can write the sequence here. So this is the TBP, right? So after TBP, the TF2. See, TF2A and TF2D and TBP, they are considered one complex only. And otherwise you can write TF2A, then TF here you can write 2, right? Because they are talking about the mRNA, right? TF2A, then TF2B, right? Then TF2F. See here, and TF2 comes along with the RNA polymerase. So keep in mind. Then sometimes they are asked actually. So then TF2F, right? And after TF2F, then TF2E, then TF2H, right? See, and this TF2H here, it is shown this small protein, but believe me friends, this is very large protein actually. This is almost equal to the protein of RNA polymerase, equal to the size of RNA polymerase 2. This is very large protein actually, right? Anyway, so this is TF2H comes at last. And what happens when the TF2H comes, then the promoter site where the entire complex has been bound, they start melting, right? And we know that approximately 14 base pair they melted over there, right? That is from the plus 3 to minus 11, right? So plus 3 to minus 11 sequence has been melted out, right? And once melting takes place, right, I mean melting means the the template strand as well as the non-template strand has been formed, and according to the template strand then RNA synthesis takes place. So TF2H, the function of TF2H is the promoter melting and using as a helicase activity, right? It acts as, see what happens here, many aromatic amino acids are there, or the amino acids having very big side chains are there, and they somehow pull the non-template strand, and they are not able to, so that they cannot be annealed together, so they work as helicase, right? I mean they are having similar activity to the helicase, and apart from that they also have the DNA repair activity. This is very important, right? The TF2H, they also have the DNA repair activity, and they work in the MMR, mismatch repair, they work actually, right? TF2H. Alright, apart from that, other transcription factor function has also been written here, and whenever we use, we come to this table again, right? So now we come back to the question. So here the correct answer you can now tell me. So binding of the RNA polymerase to the promoter sequence, no, not at all. Promoter clearance, see what promoter clearance is actually when the melting takes place, when the open conformation or open complex has been formed, what we have seen here, right? So melting takes place, so open complex has been formed, right? Open complex. So from the closed complex to the open complex has been formed. So once the open complex has been formed, many more proteins, many more factors which are responsible for the transcription initiation has been fallen down, okay? So many of them are fallen down, right? So fall of many protein, right? Many factor, fall of many factors, many proteins, right? So this is called promoter clearance, right? This is called promoter clearance. Promoter clearance. So here the promoter clearance is written, so this may be the correct answer. Recruitment of TF2D, no. This is the last transcription factor. So open promoter complex formation, okay. Open promoter complex formation, see the closed conformation to the open conformation they form here, right? But this is open promoter complex formation, this is somehow not suitable word here, right? So the promoter clearance is more suitable here, so the correct answer is 2. Alright, yeah, double but, yes, good afternoon, double. Yeah, so that's the question. Okay, now we come to the another question. This is again very good question. So first of all, we read the question, the C section question. So in order to study the transcription factor TF2H, again H they are talking about, right? So there's a last transcription factor, keep in mind. They also act on the MMR repair mechanism, they work, they comes, right? If some mismatching is there, right? Then okay, so it was cloned from a large number of the human subjects, okay, alright. Surprisingly, the subjects have mutation in the TF2H, okay, alright. Subject have mutation in the TF2H gene, okay. Also showed defects in the DNA repair system, okay. They are having DNA repair system problem, right? So of course, as we discussed, the TF2H have MMR activity also, I mean MMR mismatch repair they are having also, right? So now the things are what they are saying, some explanations are there. So DNA damage is always associated with the transcription inhibition. Not always actually, this is not always, so it cannot be correct actually, not always. So TF2H has no role in DNA repair. No, they are having role in the DNA repair mechanism, and they have repair mechanism in the mismatch repair. Okay, In mammalian system, TF2H plays an active role in the transcription coupled DNA repair process. This is correct actually. So during the transcription, if mismatch repair is there, that mismatch repair has been corrected by the TF2H. Alright. This is very important transcription factor. So this seems correct. Then D number, because of mutation in TF2H, transcription initiation is inhibited. Okay, transcription initiation is inhibited, yeah. And incompletely synthesized mRNA remains attached to the template DNA, leading to the DNA damage. No, remains attached to the template because of the mutation in the TF2H. Okay, transcription initiation is inhibited. Transcription initiation is almost completed here actually. Once the TF2H comes, almost, this is last transcription factor which is responsible for the initiation, right? So almost completed. So transcription initiation is inhibited in the last step, we can say. And incompletely synthesized mRNA, see, mRNA is not synthesized till here. This is just the conversion of the closed complex to the open complex, right? And now the elongation will start. So here no mRNA has been synthesized at all. You know, maximum 10 or 11 nucleotides has been synthesized if they enters into the open complex, and here they are not actually entering into the open complex, right? Because the TF2H is mutated. So this is also not correct. So here C number is the most correct, or only correct we can say. So 2 number is the correct answer. Anjali is saying C. Which number, Anjali? You can write the number also so that I can. Okay. All right. So this is very good question. Now 11 number question. This is again good question, we can say. The non-enzymatic viral protein X has found to be inducing a cellular gene promoter activity. Okay, so X is a protein which is having promoter activity. They show cellular gene promoter activity, right? How? तो आगे बोल रहा है although no in vitro DNA binding activity could be identified. Okay, no in vitro DNA binding activity could be identified. Okay, so they cannot directly bind with the DNA. That's what they are telling, right? So factor X or protein X is not directly bind to the DNA, but they anyhow controls the promoter activity. Okay, it was found to be co-recruited on the cellular promoter along with the cellular transcription factor. Okay, they are co-recruited, not directly in vivo. Which one of the following statements seems to be the best interpretation of the above finding? Okay, all right. Okay, so interpretations are X is a DNA binding protein. No, this is not correct because they are earlier they mentioned in the question itself. So they are not directly bind to the DNA or promoter. So this is wrong. Okay, first number is the wrong. Second, X physically interacts with the transcription factor. Yeah, we can say because they are having the promoter activity also, right? So since they have promoter activity, so we can say they can interact physically. And other thing is that they are not directly binds to the promoter. So indirectly, by interacting with the transcription factor, they can affect the promoter. So this may be correct. Okay, third is X modifies the chromatin for the transcription activation. Okay, since they are not acting directly on the DNA, so they cannot modify actually chromatin in the transcription activation. And transcription activation, I don't think the chromatin activation is required here. So this is wrong. Anyway, this is wrong statement. And X is a chaperone. No, this is also wrong. So here the second number is the most correct answer here. 11-2. Yeah. Okay, Dhaval. Yeah, you are correct. 11-2. Yes, this is the best suitable answer here. This is a good question we can consider. Yeah. Okay, now my friends, this is a big question but very good question. and simple question also. Okay, so first of all, we should read the question. In mammals, GC rich sequences are usually methylated. Okay, GC rich sequence methylated at C by DCM methylase. Okay, DAM and DCM, two methylases are there, right? Here I am writing DAM also. So they modify at the adenine, and here the cytosine. Okay, so they are talking about the DCM, which is a way for making the gene silencing. Okay, maybe, yeah, this is one of the tool for making the gene silenced. Okay, although the promoter of the housekeeping genes are often associated with the CPG island, yeah, CPG island is there, yet they are expressed in mammals. Okay, so housekeeping genes means they are expressing 24/7. Okay, and they are having CPG island also, and they are expressing in the mammals. I mean, they want to be methylated. They wanted to say here, they're trying to convey they are not methylated, or if methylated, not in the gene, maybe. So which one of the following is best explanation of it? Okay, good question. So first, methylation of cytosine does not prevent the binding of the RNA polymerase 2 with the promoter. Okay, so housekeeping genes are expressed. Okay, yeah, you can also solve with me. Okay, so this is not the correct answer. It won't seems correct. Okay, क्यों? क्योंकि methylation of cytosine does not prevent the binding of the RNA polymerase 2. Nothing like that. ऐसी कुछ बात नहीं है, right? So housekeeping genes are expressed. No. So during housekeeping gene expression, the enzyme methyl transferase is temporarily silenced. Nothing like that. ऐसा कुछ भी नहीं है. No silencing during the time of gene expression is there by miRNA, and thus shutting down the global methylation. Nothing like that. Methylation cannot be stopped globally actually, right? This is a very important feature of the cell. Okay, so then third number, unlike within the coding regions of the gene, CG rich sequences present in the promoters of the active gene are usually not methylated. Okay, what they are saying, unlike within the coding region of the genes, okay, CG rich sequences present in the promoter. Okay, yeah, so they are not there in the coding region. So methylation is not there in the coding region, but they are present in the promoters, okay, of the active gene are usually not methyl. Okay, this seems correct answer. Okay, unlike coding region of the genes, CG rich sequences present in the promoters of the active regions are usually not, then only it will be expressed. Okay, if the promoter is methylated, then it is very difficult for them to express, right? Or start the transcription and then translation. Okay, so this seems correct, but we have to read the fourth number also. As soon as the cytosine is methylated in the promoter region, okay, the enzyme of the DNA repair pathway removes the methyl group, thereby ensuring the gene expression. No, nothing like. As soon as the cytosine is methylated, the promoter region, the enzyme of the DNA repair pathway removes the methyl group. And thereby ensuring the, no, they won't remove, and they won't, I mean, if promoter is methylated, then it would be a very big problem for the genes to express themselves, right? So here the correct answer is C. I mean, third number is the correct answer. Okay, so this number is the correct answer. Okay, this is good question, analytical question you can say. 13 number question. So 2017 they asked. So scientists usually find difficulty in identifying the extract transcription termination site in eukaryotes because, okay, this is a very good question actually. See, before solving this question, we should at least see this figure. So what is this here? This is the RNA polymerase 3. This is RNA pol 3, right? And here many more factors. This is the eukaryotic system. Eukaryotic RNA polymerase 2 you see here, right? RNA polymerase 2. This is RNA polymerase 2. And here the transcript is there. They are inside the nucleus. And here two factors, so two proteins CPSF and CSTF is there, right, which will bind to the poly A signal sequence. See, poly A comes after that, but these two sequences are the signal sequence which is upstream to the poly A site, right, poly A site के upstream में ये है, तो poly A signaling sequences in the mRNA, and at that site CSTF and as well as the CPSF will bind. Okay. Now what happens actually, and in the eukaryotes certainly the mRNA has to come out for the translation, so no doubt here mRNA is not there, nothing is there, I mean ribosomes are not there on the mRNA, right? So the first problem is that the RNA polymerase is attached here only, and they got the information, I mean these two proteins, they got the clue that now it is supposed to be over, because they have to have bind at the poly A signal site, right? So poly A suppose comes here, right, but they bind here, I mean earlier they bind here CPSF as well as the CSTF, right, and apart from that they what do actually, they cleave the site, I mean just after the poly A signal sequences. So the CPSF is cleavage of the polyadenylation specificity factor, cleavage and polyadenylation specificity factor. So these protein binding at the signal sequences, poly A signal sequences, means that they have to cut the mRNA after some time, right? So before, I mean the RNA polymerase is attached itself here, I mean it is still attached here, but the mRNA has to be chopped off, right? So without getting the clue of the termination sequence it has been terminated. So CSTF is cleavage stimulation factor, right? So now you come back to the question. So the scientists usually find difficulty in identifying the extract transcription termination site in eukaryotes, because exact transcription termination site in the eukaryotes, because immediately following the termination of the transcription the 3' is adenylated. Okay, this is not correct. 3' end is generated by cleavage prior to the actual termination of the transcription. So this is correct, that we discussed, right? So 3' end is generated by the cleavage prior to the actual termination of the transcription. And third choice, the poly A binding protein present at 3' end, okay, of the transcript hides the termination. No, not exactly, right? 3' end of the transcript is complexed with 5' end for the initiation of transcription. No, this is also not correct. So the correct answer is second number. Second number is the correct answer. Yeah, 12-4, Anjali. Yeah, you are correct. 12-4. No, 12-3 is the correct answer. Okay. So 13, second number is the correct answer. Yeah, 12. Okay. So this we have seen. This is again important. Okay. So now the 14 number question. The regulatory elements of expression of ribosomal mRNA gene resides in the, okay, the choices are transcribed spacer region, non-transcribed spacer region, 5' flanking region of the individual ribosomes, and internal region of the. So first of all we try to understand these terminologies actually. See, this is the gene for the mRNA. Suppose this is 18S rRNA, this is 5.8S, this is 28S rRNA, right? So here this is the promoter, I mean this is the site of promoter. Here suppose this is the promoter site, right? Here promoter is there. Right, and upstream to the promoter is the NTS, and NTS is nothing but the non-transcribed spacer region, right? Non-transcribed. So this is not transcribed, but they have the regulatory activity on the synthesis of the many rRNA. Apart from that, many other sequences like external transcribed, I mean this one ETS, this is external transcribed spacer, right? This is non-transcribed, but this is transcribed but external. This is external, I mean this is the gene, this is the cistron, and this is the externally transcribed spacer. Apart from that, another spacer that is called ITS is also there. ITS is internal transcribed spacer, is also there between the two cistron, right? So these are the different sequences which are having regulation activity on the synthesis of rRNA. Okay, तो अब यहाँ पे choices are transcribed spacer region, the regulatory element of the for the expression of ribosomal rRNA resides in. So transcribed spacer region, first choice. So transcribed spacer region is not there. This is external transcribed region or non-transcribed region or internal transcribed region, we can say, right? So this is not correct. Non-transcribed spacer region, this seems correct. Okay. Then 5' flanking region of the individual ribosomes in, no, not individual. See, this is 5' flanking region, no doubt, but they are not individual, right? So this is also wrong. Then internal region within the genes, not always, right? Not always. So this is also wrong. So the second number is the correct answer here. All right. Second is the correct answer. Okay. See, this is the promoter. I mean, two different kind of promoters are there for the synthesis of rRNA. This is the core promoter, and this is the upstream control element UCE. This is another part of the promoter, and here the protein UBF and SL1 will bind to regulate the protein, I mean to regulate the transcription. Alright, okay, this is the next question, and the question is tRNA genes in the gram-positive bacteria were sequenced, okay, alright, while some of the genes possessed sequence corresponding to the CCA end of the tRNA, other did not, okay, some of them are not having. Interestingly, even the genes that lack the sequences corresponding to CCA end of the tRNA were found to code for the functional tRNA. This is because, right, this is because, so before solving this question we should see this figure, right? See, this is the primary transcript of the tRNA, right? And here two cleavage takes place, 5' cleavage and 3' cleavage. Here these two arrows are there. From here and here cleavage takes place, and after the cleavage here the CCA addition takes place, right? So here the CCA has been added at the 3' end, okay? And finally the mature tRNA, they are having 3' CCA having OH group, right? So after splicing and other things, so they are the spliced out this region, okay? So now we come back to the question. So the question is, the first choice is CCA end of the tRNA is not essentially to function. No, they are functional, so this is not correct. The genes that do not possess sequence corresponding to the CCA ends of the tRNA are repaired prior to the transcription. No, they are not repaired actually, okay? They are what we can say, they are added, not repaired. So C number, CCA end is added to the tRNA during the maturation. So this may be the correct answer. The primary transcript of the gene lacking sequences corresponding to the CCA and are subjected to G mediated editing. No, this is not correct. So C number is the most correct answer here, right? So C is the correct answer here, right? So friends, that's it, and we had discussion about 15 here and 6-7 questions there, so 21-22 questions are there. So this covers almost all questions they asked till June 2019, okay? So I hope this would be useful, and again I am extremely sorry for the many interruptions are there in between because of the poor internet connectivity. So thank you very much for watching, and I hope it would be useful for you, and if you find this is useful, then please share with your friends. So thank you very much.